In this section, we are showing you how to collect oral fluid specimens, also known as saliva. The collection procedure is standard, using a mouth swab, but there are options for how the sample is analysed. You may be asked to use the DDS reader, which provides a point of collection test, or POCT result, with a printout. There is a similar POCT, the DDSV, where you have to read the result on the cartridge. There is a back-to-lab option where the sample is divided into two portions and returned for laboratory analysis. This is always used if the result from either of the two previous options is not negative, or if there are any problems with the cartridges or reader. The result of any oral fluid test can be affected by what the donor has had in their mouth immediately before the test. This covers food, drink, medicines, and anything else that might stimulate or restrict the normal flow of saliva. For this reason, you must keep the donor under observation for 10 minutes prior to taking a sample, to make sure that their mouth has been empty during this period. The oral fluid collection kit will always contain a mouth swab, also called a collector, in sterile packaging. The oral fluid collected needs to be mixed with the buffer solution provided. The mouth swab has an indicator to show when sufficient sample has been absorbed. This may take between 2 and 5 minutes, with the donor actively wiping the swab around their mouth, including gums and cheeks. The volume indicator must change colour before you accept the sample. The POCT kits contain a cartridge which is used for the analysis. Transferring the solution from the buffer bottle to the cartridge has to be done carefully. The buffer bottle must be held vertically to ensure that each drop is large enough, but must be added 1 drop at a time to avoid adding too much. Too little or too much will prevent the POCT analysis. Oral fluid is a potential source of viral infections, so gloves must be worn when handling the used swab. Gloves and any other waste from the test process must be disposed of as clinical waste, not left in general waste bins on site. Although no special facilities are required, privacy is necessary and the area used must be clean and uncluttered. Specimen collection. Remember the principles that you have to follow. You are starting the chain of custody for the specimen and need to make sure that you can verify that it is a genuine specimen from a particular individual. You have to have the donor in your presence for 10 minutes before you ask for them to put the swab in their mouth. You can use this time to explain the procedures to the donor and start to complete the paperwork. By encouraging the donor to talk to you, you will notice if they are chewing gum or have anything in their mouth. We will now take you through a specimen collection using the DDS reader, followed by the back-to-lab option. The DDS should be used in the case in which it comes, which ensures a flat and steady base. The case contains the reader, printer, battery charger, and mains leads, quality control cartridges, SYS and QC, manual and troubleshooting guide. Before you begin using the DDS machine, have the manual to hand. Check battery status of instrument. Check that the correct drug test panel is applied, DDS6. You may have altered the settings without noticing. Check time and date are correct. Check the printer is connected via the interface to the reader. Conduct the SYS and QC checks to ensure the DDS instrument is working prior to use. Testing must only be carried out if both cartridges pass. If either fails, do not use the DDS, but move straight to the back to the laboratory for testing. In addition to the DDS, you need the collection kit, which contains the swab, buffer solution, and cartridge. You need a chain of custody form, your checklist, the donor information sheet, current medication questions, and back-to-lab kit, in case the result needs laboratory analysis, and any customer special instructions. Ask the donor to read the donor information sheet and then explain the use of the checklist. The donor must follow the checklist with you, so talk them through it, continually referring to the numbered steps, explaining what you are doing at each point. This will take you through the whole process in a logical way and will minimize the possibility of you making mistakes. Follow the checklist and complete the first parts of the chain of custody form. Once the 10-minute observation period is over, invite the donor to choose a kit and move the remainder out of your way. Enter the product reference, lot number, and expiry dates where indicated on the form. Put your gloves on, open the kit, and ask the donor to remove the swab from the packaging and place it in their mouth. Encourage them to swab around their mouth until the whole of the indicator turns blue. This may take 2 to 5 minutes. Ask the donor to hand you the swab. Place it in the buffer bottle, swab end first. The oral fluid is mixed with the buffer by shaking it for about 30 seconds. Remove the test cartridge from its packaging and check the silica gel is the correct color. If not, discard the cartridge and ask the donor to choose another kit to get another cartridge. Invert the buffer bottle vertically over the small well of the test cartridge and add 1 drop per strip, allowing each drop to be absorbed before applying the next. If the dye in one or more of the strips has not run through to the main well, 1 extra drop can be added in the small well to that strip. Once all 4 lanes of the main well begin to run, choose Begin New Test from the main menu on the reader. Confirm that the DDS6 panel is set. Fully insert the test cartridge into the reader and press Enter. Note the start time where indicated on the form. The results will be displayed on the reader and a printout will be produced automatically. If additional printouts are required, hold down the Down key when prompted by the reader. Note the read time on the form. If the result is negative, complete the chain of custody form by ticking negative. The donor and you should both sign the printouts and chain of custody form. If the DDS screen result is not negative for any of the drugs being tested, explain to the donor that you will need a further specimen to send to the laboratory for analysis, and you will need to take them through a few further steps to ensure the security of the specimen. Invite the donor to choose a back-to-lab kit. This contains a swab, buffer bottle, and 2 vials. Put on fresh gloves and remove collector and buffer from bag. Confirm with the donor that both are in date. Remember the principles that you have to follow. You are starting the chain of custody for the specimen and need to make sure that you can verify that it is a genuine specimen from a particular individual. You have to have the donor in your presence for 10 minutes before you ask for them to put the swab in their mouth. You can use this time to explain the procedures to the donor and start to complete the paperwork. By encouraging the donor to talk to you, you will notice if they are chewing gum or have anything in their mouth. When the indicator shows sufficient sample has been collected, place in the buffer bottle and shake vigorously for 30 seconds. Then, separate the buffer mixture equally into the 2 specimen containers, ensuring that each contains the minimum amount indicated. Ask the donor to initial the seals and then place 1 over each container cap. Complete the chain of custody form, carefully following the steps on the checklist. Use the current medication questions and make sure all the boxes on the chain of custody are completed correctly. Ask the donor to read the consent statement and sign the form. If this is not signed, we cannot analyze the sample. Put the 2 sealed specimen containers in the pocket of the chain of custody bag and the top copy of the form in the other pocket of the bag and seal the bag. Give the donor their copy of the form and let them leave. All remaining paperwork must be dealt with according to your instructions. DDSV. The DDSV is a visual test with no reader or printer, so the test results are interpreted by the human eye. Like the DDS, it is a screen test only, and any not negative results will require laboratory confirmation. Collect the specimen as previously shown. When you have the specimen in the buffer bottle, remove the test cartridge from the packaging, being careful not to touch the strips of the cartridge. Check the silica gel is the correct color. Invert the buffer bottle vertically over the small well of the test cartridge and add 1 drop per strip, allowing each drop to be absorbed before applying the next. If the dye in one or more of the strips has not run through to the main well, 1 extra drop can be added in the small well to that strip. Apply the Windicator onto the test cartridge, ensuring that it is the correct way round. The test cartridge should not be read until 5 minutes after the application of the specimen, but within 10 minutes. The absence of any test lines indicates a not negative result. The appearance of a line, however faint, indicates a negative result. If the DDSV screen result is not negative, invite the donor to choose a back to lab kit.